that's clear. The quirks are like, I wanted to revisit one thing of the person that was asking before about the optogenetic for individual slice. That was the spines. That was the last questions. I just want to comment and add something that is also, I find quite cool, that you can use also lasers to basically like uncage molecules. That means like, use a laser or a light to release specific molecules close to synapses. This, for example, glutamate uncaging is something that has been used for a long time to induce not a direct optogenetic activation of a synapse, but to induce the chemical activation of the synapse through a endogenous means, which would be like you induce glutamate to be active at that specific synapse using light. So it's also a different type of, you can be quite creative. And at the moment, there is quite a few labs in the world that are developing not only a fluorescent sensor, like the one that we discussed on Tuesday, but also molecules that can respond to laser power or to light to do a lot of interesting stuff. Okay, so we move now to a very interesting and historically nice lecture. That is, I chose it as sort of a case study that can really show you and illustrate how you go from asking a very important question about functions of the brain down to the cellular plus molecular details of this. So of how this phenomenon is created in the brain. So sort of a case study of what system neuroscience would do. So in the end, I want to talk about how the brain creates, stores, and recalls memories of personal experiences. And these are what are categorized as episodic memories. So before we go further in the details of how this was discovered and how actually the brain does it, I want to clear the air and define what I mean by episodic memories. And Wilde, don't laugh about this. So episodic memories are memories of things that happens to you during your lifetime. For example, you've never been in Norway, in Trondheim, on the 17th of May. It is a day of a national holiday of the Constitution, I guess. Wilde, you can nod if it's correct. And they create parades in Trondheim in which people that belong to an association decides to, go together and march together through the parade. And in particular, there is one group that is very Star Wars aficionados that go around the city dressed like people and character from Star Wars. So it might happen that you are in Trondheim on the 17th of May and you bump into Darth Vader and the stormtroopers. And this was actually a picture that I took. This is the first time that I saw them, and it's very etched and engraved in my mind. This is an experience of something that has happened to me, so in which I live it as a first person, of an episode that has a clear definition in a place like Trondheim and a time like 4 years ago. So this is what we define as episodic memories. And the idea, the question that I want to try to answer today is, how are memories like these encoded in our brain? And one of the first definitions was that memories like these are thought to be encoded in the brain as some enduring and long-lasting physical changes in the circuit of the brain, so that when you take and you reactivate that long-lasting physical change, which is called in jargon engram, you can be able to reactivate the process of remembering that particular memory. In a more scientifically put terminology, Richard Semon came up with the definition of engram in the beginning of the 20th century. And he defines that an engram, which is what corresponding to the memory trace that we talked about before, is elastic change in the brain state and structure, both so at the level of synapses forms, at the level of activity patterns, that occur in response to an event on experience, that goes dormant when that event of experience is not present anymore, but it can be awakened if we present either parts or the whole original sensory stimulus that were presented during the event. For example, when I watch Star Wars, I will remember that the 17th of May, there is a crazy person going around in Trondheim dressed as Darth Vader. The process of reactivating that dormant memory trace is called ekphory. Now we are going to call this reactivation of the dormant memory trace retrieval or recall. So these are the two terms that we're going to use today. So in the end, he came up with 4 criterion. So we know that memories induce a change in the brain. That can be either a change of state, that is a change of activity, or a physical change, that can be a change of connectivity between neurons. How can we define if a change that is in the brain is actually an engram for a specific memory or not? Simon came up with 4 different criteria to satisfy. First of all, that change must be specific to that event or that memory. So you cannot have, or let's say, in this definition of events of a specific of an episodic memory, the specificity is actually quite an important criteria. It has the potential to be retrieved by naturalistic cues, meaning that what Simon said at the end, if you provide the same sensory stimulus or reminder sensory stimulus that might make me remember of the moment in which I saw the parade in front of them, I should be able to reactivate that memory trace and to the activation induce the recall itself. It should be consistent, this memory trace, between encoding and retrieval, because it should be the same type of memory trace that is basically present in our brain. And it should be active during encoding, active during retrieval, but dormant between encoding and retrieval. Because otherwise, it would be active during encoding and retrieval, but you don't actually remember what that memory trace should be encoding. It's definitely something that it's not a specific connection between that specific physical substrate and the remembering of that memory. So in the end, specificity, the potential to be activated by naturalistic cues, the potential to be consistent between the moment in which the memory trace is formed and the memory trace is reactivated, and should be existing in a dormant state between encoding and retrieval. These are 4 criteria that people have used to guide their search of the engram and their search for those physical changes in which memories are encoded. So if I ask you, who have a very strong molecular background, what form an engram could take, you say that the physical changes associated to engram formation might span a different and multiple realm of biology. They could be, memories could be encoded in the pattern of gene expression that, for example, neurons express during their lifetime. It could be present at the level of synapses. It could be embodied by the potentiation of some synapses, the repression of others, or new synapses that are formed in response to experience. It could be encoded at the level of individual neurons, for example, deciding to make a new dendritic branch or not, or to connect to another neurons or not. It could be embodied at the level of population of neurons, namely in the decision of which neurons are active at the same time, are active together at a specific time. Or it could be present encoded at a level of a network of neurons. In every node in this graph is a brain region. You might imagine that an engram might consist in the synchronous reactivation of areas of the brain, like, for example, in this case, the prelimbic cortex, the anterior cingulate cortex, and the lateral amygdala. In the end, the physical trace of a memory could be at every one of these levels, from the most microscopic to the most macroscopic one that you can find in the brain. And most likely, it is in all of them at the same time, namely that you have different expression of genes that induce potentiation of depotentiation of synapses, which induce change the way in which a neuron is active or connected to other neurons, which influence which cells in one area are active at the same time, which again, it's not something that you find all over the brain, but you find in specific areas who take care of creating memories, of building memories, or of storing information. So one thing that I want you to have clear in mind is that when we talk to the engram today, we're going to be looking at the population level, but the physical changes that are associated to memory formation span mostly all the different layers of complexity of the brain, from genes to networks. Okay. One of the first person that actually really tackled the question after Simon of what type of form a memory could take in the brain and an engram could take in the brain is the beloved Donald Hebb that we already saw previously on Tuesday, who in that moment, with that treaty that was published in 1949, the Organization of Behavior, he put forward that idea that if two cells fire at the same time, the connectivity between them can be strengthened. And strengthening the connectivity between these two neurons is able to create a group of neurons, he called neuronal ensembles, that are able to be active at the same time and encode the same type of information. So the idea that he put forward that in the end, the process of creating a memory is basically based on selective which memories, which neurons are able to be active at the same time, to be co-active, and so that those neurons will form a neuronal ensembles in which the information about that specific experience is actually encoded. So if you want, the neuronal ensemble that Hebb has defined and the word engram that Simon had put forward are sort of, in this exception, synonymous of the same thing. A group of neurons that is coactive, a group of neurons that encode a similar information. This, of course, brings also to what he also mentioned before, that integrating the idea of Simons, the idea is that an engram is basically the information about a specific event is able to be etched, engraved, or written within the activity of group of neurons that were active during the experience itself. The connectivity between this group of neurons gets stronger and stronger according to the Hebbian hypothesis of coactivity inducing LTP. So during a process that is known as consolidation, so that in the end, if you repropose part of the sensory inputs that were present during acquisition, you are able to induce the retrieval of the memory, that is basically the reactivation of that population of neurons that was active during acquisition, in a way that allows you to remember the original experience. And this phenomenon goes on and on and on. Those neurons will be reactivated when you remember the experience. They will go back to a form of dormancy when you don't remember it anymore, to be ready to be reactivated when, again, you induce a sensory stimulation that recapitulates acquisition, and so on and so forth. There is an interesting phenomenon. There is the phenomenon of reconsolidation that we are not going to go into. But if you're curious, let's just talk to Maria when she's available, because she has been one that has been studied this in the lab quite extensively. And also, let's discuss later in the discussion. So the hypothesis that comes from the idea that basically Simons and Hebb put forward is that experience induces a physical change in the brain. This physical change is embodied in the ability of a group of neurons to become coactive and to encode information about that specific experience, so that the reactivation of that particular group of neurons that we call the engram is able to induce the retrieval or the recall of the memory of the specific event that has created the engram itself. So one of the questions that came after what type of form the engram could take is what could be the engram located in the brain? And this type of question was faced with the means that we have discovered together today, that is basically lesion experiments that Carl Lashley put forward to understand where the memory of a specific experience was actually located in the brain. And the conclusion were actually quite interesting, because himself said that the trick was not to fire where the memory trace or the engram is, but where it is not. Why? Because he did an experiment in which he trained rats to solve mazes, and you could be astonished by how many answers you can get by simply teaching an animal like a rodent to learn to find a goal, right, and motivate it with food. But what he did, he basically trained these rats to locate a maze or to find a way to a reward in a maze, and then he went and lesioned individual portion of the brain of the animal to understand which lesion was able to impair the performance of the animal. So if you lesion one area that is important for storing the memory of that maze, your prediction will be that the animal is not able to find the maze anymore. And what he found is that there was not one specific area of the brain, he was looking at cortex at the time, that was alone responsible for impairing the memory. But they do needed a very large lesions, or let's say lesions that would cover multiple type of brain area to induce actually an effect. So it was the size of the of the lesion of the cortex that was correlating with performance of the rat, and not the specific location of the lesion. Today, we know that this concept was not entirely true because we know that there are specific areas of the brain that when you inactivate, you impair the ability of the animal to actually navigate the maze. But it clearly highlights one important principle of brain function, and it is that memory is not localized to a single brain region, but is actually distributed into the activity of many neurons that are located in many different brain areas. Not one single brain area works in isolation. We will see later how even memory is able to basically be influenced by the concerted activation of the hippocampus and the neuronal cortex, the prefrontal areas of cortex, and another area of cortex that is the amygdala. One caveat of this expression here is that with this experiment is that Lashley was doing lesion and waiting for the lesion to recover. By the time that the lesion and the animal had recovered, the brain had already compensated the loss of that specific area if the lesion was quite small, so that the animal would not see an impairment in the performance in navigating the maze. If he was doing an acute experiment, he would have found something, some meaningful region early on. But let's keep it for the next part of the talk. However, there was a big, so Lashley arrives to the conclusion that the memories are not located in individual areas, but they're distributed somehow everywhere in the brain, everywhere in cortex. And you cannot really find one specific location, but you need to take out multiple of them in order to find the vision. However, Wilder Penfield was a neurosurgeon that was, again, looking for epileptic foci in human patients and was putting electrode to record from the areas of these patients. What he discovered is that when he was doing some experiment with this patient, and instead of just recording, was inducing a little bit of electrical stimulation to stimulate one specific part of the area, one specific part of the brain, sorry, when he was recording, that was the lateral superior temporal lobe, he was able to induce the patient to recall an experience that had happened in the past. So if you stimulate something in the brain, in that specific area, and you ask the patient, what are you thinking right now? What is going on? The patient were responding, yeah, I'm thinking about the moment this morning when I was putting on my shoes to come here and come here to the hospital. And this was literally what he was saying. He was able to identify loci in the brain of the patients where the electrical stimulation was not able to induce sensation, but was able to recall vivid experiences that had happened in the past to the patient itself. So in the end, this goes a little bit in contrast to what Lashley was saying. Lashley was saying, you know, the memory is not in a specific place, it's somewhere else all over the brain. And Penfield instead was giving the answer, no, there are some specific areas that when you stimulate this area, you're actually able to elicit memory response. So let's start again. And the thing was sort of reset at the beginning of the 50s. The experiments were actually inconclusive, and people could not really look and see where the physical traces of memories were. That's where HM came and revolutionized the concept of memory study and our understanding of how memory processes work in the brain. Again, I don't need to talk about Henry Molaison anymore because you know him by heart, probably better than Brenda Milner, but she was the psychologist who was working with Scoville, who was a neurosurgeon, to try to understand the impairment that Henry Molaison had. And she immediately understood that this was an impairment in the ability to produce new memories of personal experience. And most importantly, he was not impaired for a very short period of time. He could remember something for a few hours, but then after those few hours, memories would be lost. So she was the first one, together with the other co-workers that she worked on, to suggest that the structure in the medial temporal lobe are necessary for the formation of a memory trace that is able to last long and become basically a long-lasting memory in the brain of humans. And that really brought to sort of a breakthrough and the new beginning of the trying to understand how memory traces and how memory works in the brain. It took the work of many researchers over decades of time to actually arrive to the conclusion that memories are encoded in the ensemble. To actually put forward an hypothesis, let's go there, that the sparse population of neurons that are active during an experience encodes the memory of that specific experience and is reactivated when that specific experience is remembered. This is what Simons had put forward as the engram. This is what Hebb had put forward as the cell assembly. And starting from this hypothesis, we move fast forward to the end of the 90s, where we have the introduction of some knowledge and some technologies that allow us not only to locate the locations where the specific memory traces are, but even to understand a little bit more about mechanisms that lead to the formation of these memory traces and to the process by which these memory traces are consolidated in long-term memories and reactivated when recall happens. The game changers in this quest to understand how the brain works are basically two. On one hand, it was the discovery that a fraction of the cells that are active during an experience start to express a series of genes that have been called immediate early genes. Part of the immediate early genes are CFOS, ARC, ZIF, Homer, and many others, whose induction is basically connected to the ability of this neuron either to be activated by experience or to become plastic following an experience itself. So these genes became a tool to be able to label selectively the population of neurons that were activated during experience, which in our hypothesis were Simons engram. And how? Because we can combine the construct that I was telling you before, that is the ability to induce the expression of a specific protein with the protein the expression of a specific construct with one of the promoters of these immediate early genes, and to activate the recombination only in moments where we inject tamoxifen. So by using the combination of a promoter for one of the immediate early genes, and the most important was FOS, and the other one, and the CRE-RT2 construct, we are able to induce the expression of a payload protein, in this case, just the fluorescent reported mCherry, only in neurons that are active at a certain period of time in which we give the ligand that is tamoxifen. Neurons that do not have activity at the time, or they are active when there is no tamoxifen, are not able to express our reporter. So in the end, by combining a genetic promoter for the expression of a recombinant protein, of recombinase, only in the cells that are active during the experience, we are able to trap neurons that were active at one specific moment in time, which is the moment in which the memory is formed. This is really probably one of the most important advancements for this field, and the technological advancement that made the discovery and all the things that I'm telling you in the second part of the presentation and the papers that we're going to discuss this afternoon possible. Because at that moment, when you have that the neurons that are active during a specific period of time, that is during experience, are actually genetically accessible to us, we can induce this way to trap this neuron and have them express some recombinant protein, like for example mCherry. But most importantly, we can use this technology to have only the neuron active at the moment of acquisition express effector proteins like GCAMP, like channelrhodopsin, like the DREAD receptor for chemogenetics, which allow us the possibility to study their activity and to manipulate their activity in a controlled fashion, to basically test the hypothesis that Simons and Hebb had helped put forward. That is, that the memory of the experience is actually encoded in part of the neurons that are active during that experience itself. Is that clear? So this application of these two technology was actually very much making us able to now study the behavior of animals, and in particular rodents and in particular mice, because these you need to induce via transgenesis, which is easier in mice, and you will see next week why and how it happens, and to study them when animals are able to form memories. And one of the most, let's say, basic type of memory that you can create in a mouse is an association similar to the one that I have already introduced you when I was talking to Pavlov. You have a stimulus that is able to elicit a response. You have a stimulus that is not able to elicit a response per se. You pair the two together and are able to transfer and basically teach the animal that the stimulus, that usually would be neutral, can predict something that actually is able to use a response and test the formation of this memory by testing if the animal is able to express the experience itself. This is something that Vilde is going to talk more in the afternoon. I'm just going to highlight the principle. You basically introduce a mouse into a room, into a chamber that would be neutral. The mouse would explode and run around, and you will see how actually very comfortable they are when you put into this chamber. But at a certain point, in an unsignaled way, you give a foot shock. So the grid, the base of the room is actually electrified. So you give a foot shock that, of course, induces a pain reaction into the animal. And you will see what this looks like in the afternoon. So that when the animal will be associating that particular context is actually dangerous, and form a memory of that particular event that will express by expressing fear when the animal is put back another time in that room. At this moment, the animal starts to be fearful of the environment, even if you give no shock during the recall session. The animal will have created a specific memory for that particular environment, because if you put into another environment, which is neutral to it, it's not going to freeze. We know that there are many brain areas which are involved in the formation of this type of memories. These are few of them. You need a prefrontal cortex is involved in the formation of these memories. The hippocampal formation, which is the place where we suspect the ability to create this memory is because of the studies that came from Henry Molaison. You need the amygdala, and then you need the two areas of that are the nucleus of the striatum terminalis and the thalamus, which we're not going to talk about. But the interaction between prefrontal cortex, hippocampal formation, and amygdala is fundamental for creating a memory of the contextual fear condition. And this also highlights the process of Lashley that was not really able, like that you could see and say, okay, these are basically like different areas in the brain that are all involved in the formation of the memory processes. So the first question that came and could be resolved by this means was to understand if neurons that are active during an experience are actually the same one that are reactivated when the experience is recalled. And this is a study that happened in the end of the 90s. This was quite simple at that moment. They basically, you put the animal in a specific context that you call context A. And after a certain delay, you either re-put the animal in context A or put the animal in a novel context that has never seen before, context B. And what you do is you quantify the expression of these immediate early genes. You will have a population of neurons that will be active when the animal is put in context A. And you want to ask if this population of neuron is the same one that will be active when you put it again in context A after the delay. And similarly, if instead the population of neuron that is active when you put it in context B is a different one. This is a very ingenious way to answer this question was, since you know that the immediate early genes is first expressed into the nucleus and then translated into the cytosol, you can look at see when the either the mRNA or the protein for that particular gene is actually expressed. So if you look at if you put the animal in context A and you go immediately and look for the presence of that mRNA for that specific immediate early genes, you can see that you will find some foci in the nucleus, but nothing in the cytosol because the protein has not been translated yet. If you wait a little bit, then after the delay, the protein will not be expressed anymore in the nucleus. The gene will not be anymore in the nucleus because the animal is not; those cells are not active. The experience is over. But you can find the protein at this moment in the cytosol. So you can clearly distinguish if the neuron was active in an immediate past or in a remote past by seeing where you can find the mRNA or the protein. So what you can do is that you can put the animal in context A, wait a little bit, and see that you find a lot of genes in a lot of mRNA into the intracellular, into the nucleus of neurons in the CA1 area of the hippocampus. If you wait a little bit longer, you will find this in the cytosol. Now, if those neurons that were active the first time you expose in context A are reactivated when you put it in context A after the delay, you should find that you find both the mRNA that comes from the second exposure to context A and the protein that comes from the first exposure to context A. While if the same neurons that were active in context A are not activated when you put the animal in context B after the delay, you will look and you will find that those basically like that some neurons will have the protein in the cytosol, the one that were active in context A, and some other neurons will have instead the mRNA in context B. And this is indeed the result. So if you look at animals that have gone first exposure in context A, then delay, and then second exposure in context A, you find a lot of neurons that have both intranuclear foci and cytoplasmatic protein. While if you put the animal first in context A and then in context B, you will find that there is a lot of neurons that have cytoplasmatic nuclei, some other cytoplasmatic protein, other neurons that will have the intranuclear foci, but they do not overlap. They focus on just one population here. Okay, so in this study, they show that indeed the cells that were active during exposure on the formation of a memory are the same neurons that are actually active when that memory is recalled. So when the animal is put again into the context A. But are these neurons the ones that carry information about context, about the first experience, or are just neurons that are active in that specific experiment for whatever reason, because there is a sensory stimulus that activates those neurons whenever the animal is in context A and context B? To answer this question, there is a new experiment that has been done that has used another manipulation technique that we have used today. That is, you basically train in another area of the brain, that is the amygdala, you're basically able to express the diphtheria receptor only in the cells that were active during the acquisition of the fear conditioning experiment. If those cells are the ones that are carrying the information and are storing the memory for that specific event, you can imagine that killing those cells will basically erase the memory of that fearful event, that is the food shock, from the mind of the mouse. How can you test that? If you basically express the diphtheria receptor into the cells that were active during the acquisition of the fear conditioning, then you kill those cells by injecting the diphtheria toxin, and then you test if the animal does or does not freeze to that specific environment. So if the animal remembers or does not remember if the environment where he was put was actually the one in which he got the shock. And what you see is that when you kill the animals, so this red bar here, in which the cells that were active during the acquisition of the memories were killed, the animal shows much less freezing response, that is the way in which we quantify fear, than an animal in which instead was basically like in which those cells were active. So you can see that if you kill the cells that were active during the moment of the acquisition of the memory is gone. The third experiment is, and this is the question that we ask, okay, this is the case, the first case, right? The loss of function type of approach. You kill the cells, you don't see the function anymore. You kill the neuron, you don't see the memory being expressed. The other question is the opposite. If you activate those neurons, are you able to induce the recall of the memory, even in an environment that normally should not be fearful? The answer is yes, and this is what came through optogenetics. They use a different method to label the active cells, which is based on doxycycline and tetrodotoxin gene promoter, but the result is the same. They are able to express channelrhodopsin, that is the effector of optogenetics, only in the neurons that were active when the memory of this experience was formed. In this case, they target the dentate gyrus of the hippocampal region. When they stimulate those cells in a context in which the animal was not being before, like it's not the context when the animal was shocked, they were able to induce an increase in freezing only in the animal in which they were stimulating the neurons that were active during acquisition of the experience. So what they had done, it was basically transfer the memory or induce the reactivation of the memory that one particular context was fearful because it was the one in which they received the food shock to a neutral context that they had never seen before. So in this case, the engram had been found. The 4 criteria of the engram, the specificity to an event or memory was actually there. Those neurons are reactivated only when you put the animal in conditions that are the condition where the memory was formed, but not in conditions that are not similar to where the memory was formed. It has the potential to be retrieved by naturalistic cues. So the cells that are active during acquisition are reactivated in the moment in which you basically put the animal back into the same context. It is consistent between encoding and retrieval because, again, it's the same subset of neurons or a subset of the neurons that were actually active at the moment of acquisition. And it exists in a dormant state between encoding and retrieval because those cells are not always active to tell the animal you should freeze. But at the same time, the activation of this neuron is sufficient to induce the behavioral response that is actually associated to that particular memory. And this really opened up to a series of studies that have dissected the way in which memories are formed, the way in which memories are created, how you can actually use a memory to guide further behavior in a way that was really unthinkable until, I would say, even 15 years ago. You will see today there are 3 different studies that we are going to be discussing today in the individual groups, in which people have been able, by using this trapping and the optogenetic stimulation technique, to create false memories, to induce the animal to believe something that was actually never there, to use positive memories and the remembering of positive memories to alleviate symptoms of depression in a mouse model for depression, something that gives a sort of I would say biological foundation to what people do in therapy. Or to change the valence of the memory that during encoding had been associated to negative outcome, like a food shock, being transformed into something that instead was associated to a positive outcome. And this is what you basically will see during the journal club this afternoon. And for people that have not got the other paper as assignment, please read them. They're quite easy to understand, and they're actually quite interesting to know how you can use technological advancement to actually dissect a lot about memory. These studies also gave back some sort of dignity to the poor Lashley, who was, who had been sort of wiped out by Penfield discovery that indeed there is small areas of the brain in which memories are discovered, because they found the discovery, one of the discoveries that came from these engram labeling methodologies is that indeed, even for one specific paradigm, like the contextual fear conditioning, that is the one that you have seen before, the engram, if you want, is distributed across many areas. I've told you before that you can stimulate engram cells that are the trap cells in the amygdala or in the hippocampus or in the prefrontal cortex, and you're able to elicit responses, the reactivation of memory, depending also on the moment when you do, and we'll see it in a moment. At the same time, it also highlighted the centrality of the hippocampus for the production of these specific personal experiences, but not for the long-term memory of these specific experiences, something that Brenda Milner had found out with patient type N. Because what one particular study of this showed that during acquisition, you create a memory trace in multiple areas of the brain involved in the phenomenon of remembering the fear conditioning experiments. You activate or create memory traces in the hippocampus, in the medial internal cortex, in the lateral amygdala, and in basolateral amygdala, and in the medial prefrontal cortex. If you want to retrieve memories at a very recent time points after memory acquisition, what will work is the reactivation of the cells, of the engram cells of the hippocampus, of the internal cortex, or of the amygdala. If you reactivate the engram into the prefrontal cortex, this is not going to be effective in inducing the memory of that specific experience. However, after the moment of the acquisition of the memory, something goes on in the brain for which information is constantly transferred from these 3 areas. And when I say constantly, it's a little bit of an overstatement, but it's transferred offline between these 3 areas. This is what we define consolidation. And one proposed mechanism is that replay phenomenon that we have seen yesterday was identified via decoding, by which information at a certain point, like the memory trace in the prefrontal cortex, gets strengthened. So much so that if you want to recall the memory at remote time points, you will be successful if you activate the medial prefrontal cortex that was unsuccessful during the beginning of at the recent time point. This goes very well in hand with the data coming from amnesic patients that show that the memory of the hippocampus is important for the creation of the of recent memories, or let's say put it like that, is important for the creation of every memory, but is important for the recall of memory that happened recently, but then becomes dispensable if you want to recall memories that happened past in time. And again, this is quite a very strict oversimplification because it might not be entirely true for all the type of memory. It depends also from the number of details, vividness, and emotional attachment, and so on and so forth. And now I think that you all, in your mind, I know that there is a burning question. that yesterday I told you that the hippocampus had a cognitive map that was mapping space. So there is a sort of a not agreement between our physiological recording that we saw yesterday, the fact that in the hippocampus we have place cells, and then we have head direction cells, border cells, speed cells, and grid cells, and the fact that the hippocampus instead is fundamental for the formation of episodic memories. So how do we solve this riddle? How do we know how does the hippocampus contribute to memory? This is still an open question, so I'm not going to give you a definitive answer, but I'm going to tell you the hypotheses that have been put forward based on different type of pieces of evidence and where we are heading with our investigation. So the first thing is that we need to deconstruct what an episodic memory is. An episodic memory is a memories of an event that occurred to yourself in a specific spatial and temporal context. If we go back to the image that you showed before, 3 different elements distinguish this particular experience. The content of what this particular experience is about, the fact that I saw Darth Vader and Stormtroopers, responding to the question, what? The spatial environment in which this memory was actually created, so the fact that it was in Trondheim, specifically Munkegata 5, and the fact that this happened on 17th of May of 2016. So in the end, you can think of an episodic memory as something that combines together 3 different axes or variables, the what, when, and where axis. These 3 elements might be the key element of an episodic memory. So what this study from Henry Molaison puts a spotlight in the medial temporal lobe, and people start to think that the medial temporal lobe, via the possibility to form a spatial context of the environment, and as we have seen from yesterday also, a temporal context through the lateral and frontal cortex, they're able to put together the where and when of an episodic memory and fill it up with the what. So based on the different observation, amnesic patients, recording of place cells in the hippocampus, 3 different hypotheses have been put forward. The first one being that the hippocampus is the seat of the cognitive map that Tolman was talking about. They had the ability, cells in the hippocampus and the entorhinal cortex have the ability to create a spatial representation that allows you to put your memories or the event that happened to you into a spatial context, and that's how you organize your knowledge in the brain. A relational theory that tells you that the hippocampus instead does not really create a map of the space, physical space around you, but it creates a relational map of the things that happen during your environment. So it's a little bit subtle as a distinction, but it does not really tells you that, you know, this is a map in the Google term and you should navigate it, but it tells you that you had an experience of that specific location that puts together, like, for example, the fact that you had a window, the fact that you have a desk, in my case here in the office, the fact that you have a computer that you're talking to, and so on. So thereby creates a memory space where information about your experience is actually put together. These 2 can sort of go into the same direction, saying the hippocampus creates sort of a memory space that can be either physical or conceptual. But the last one instead sort of goes through a departure of this and say that the hippocampus creates a code for experience that acts as an index, but that actually whose only function is the one to point to information is stored in the brain, which is basically in the location of cortex. So the idea would be that when you do an experience, you have a certain piece of information, visual information into the visual cortex, auditory information to the auditory cortex, and so on and so forth. And you use the hippocampus only to bring them together, to use it as a pointer, to know the address where you have stored different pieces of information. So the first thing that really points to a direction that the hippocampus is much more than space is that the same way and the same cells that you use to organize the space around you and to map the environment that are the place cells, you use them also to organize experience in a more abstract way. You remember yesterday we talked about the Jennifer Aniston neuron, the fact that this was encoding a concept. People now think that what actually we see as plays encoded in the activity of neuron is a way in which the hippocampus organize the experience that you have gone through. And this comes from one study in which basically in rats and one study in humans. In rats, what they did, they recorded sequences of place cells that were active when the animal was moving on a linear track, like the one that we saw yesterday. And they were able to record the same cells when the animal was performing a different task. It was basically representing a series of sweep, a series of nodes, and deciding which node the animal should press the lever to get a reward. They found that the same way in which space was organized in the activity of the play cells was also the same way in which music or tones was organized in this task, thereby putting forward the hypothesis that maybe what we call space, it's indeed a cognitive space that is not used to map specific type of specific like spatial location, but the more of a general way to organize the experience that we go into the world. The fact that we are, for example, living this in this moment, and then we live something else in the next moment, and something else in the next moment, and so on and so forth to create a uniform representation of experience. In this case, and let me just skip to the end of this because we are basically a little bit late. What we are now starting to think is that the cortex in general and the hippocampus might work in tight, close connection with each other. And what we look at the engram as an engram in the hippocampus might actually be an index that allow us, as the index hypothesis was mentioning, to connect and retrieve information that is actually stored somewhere else in the hippocampus. Sure, somewhere else than the hippocampus. The hippocampus, yes, has the ability to create map of experience. You find the place cells, and if you lesion the hippocampus, you have problems in navigation. You cannot solve the Morris water maze that Maria was talking about yesterday. But at the same time, at least part of the cells of the hippocampus might act as a pointer, as a librarian that tells to the brain, look, at that specific moment in time, you actually were subjected to this type of information, and you can find this type of information in different areas of cortex. Indeed, when we put the animal into the same room of the fear conditioning, we do not have the reactivation only of the hippocampal cells that were active at the moment of acquisition. We have a reactivation of engram cells that is general to many, many areas of the brain. But if we take out the hippocampus, so silence the hippocampus at the moment of recall, this pattern of activity, reactivation of engram cells in other areas of cortex is also lost. So you need the hippocampus to reinstate the pattern of activity that you had during acquisition in other areas of cortex. So the jury is still out there. We don't know how specifically the hippocampus contributes to memory processes. We know that we have multiple observations, that there is definitely a spatial and a temporal code in the hippocampus. We know that there is an engram code in the hippocampus. And what we thought the engram might encompass the spatial and the temporal code might actually be a different dimension in which the hippocampal works. There is the ability to index different knowledge that is stored in different areas of cortex. And that concludes this lecture. It basically propels us to our break and to the moment, if you have any question, to just ask them.
There's two.
There is in Slido, yes. So let's go to that. Can you explain déjà vu with memory engrams? This is probably like the most let's say, immediate thought that you could have, right? I think in theory you can, but it would need to be able to record from these memory engrams in humans. And in humans, you cannot really record from these cells in at the cellular level, right? You can see with the methodologies that we use in humans if an area of the brain was active, but really you don't have cellular resolution unless you have this microelectrode that are used specifically in patients. So my answer, my personal handpick could be yes, this could be like you have, for whatever reason, the animal is moving into like, reactivating the wrong ensembles at that moment. And that's why you have the feeling that this is something that you've seen before, but it's something that needs, of course, testing. And it's not very easy to test because mice cannot report déjà vu. Let's put it like that. The other point is, could the method killing a neuron losing a memory used for people treated with PTSD in the future? Yes, so there is, it would be complicated to kill the neuron and losing a memory, because one thing that I didn't tell you is that, of course, one neuron can belong to multiple memories. So it's not that, one neuron is taken to a memory and that neuron is not going to be participating in other engrams, because otherwise you would, even if the hippocampus has like really a lot of cells, you would lose the possibility to create memories at a certain point, right? You should be saturated with the threshold. one thing that you will see through the experiments that, or through the papers that you will see this afternoon, is also that you can work in different ways to extinguish, for example, the association between a specific room and the unpleasant event. Mice can learn that the environment in which they receive the shock is not harmful anymore by, for example, reactivating the memory trace in a different session in which you keep them for a very long time into the same context, that leads to the formation of a second memory trace that is the association, this place is now safe. So there are different ways of trying to manipulate neurons that are, memories that can be extended from mice to humans, but killing the neuron would not be my favorite one, just because it would be quite tricky technically and quite imprecise from the memory point of view. Any other question about these points, about how engrams work and how basically they can be used to drive further behavior? Okay, so we saw for the lunch break, we're gonna meet together here at 1 with Vilde, who is gonna tell you how we actually set up a contextual fear conditioning experiment. And you will have an assignment to do. I'm gonna be here as well to tell you like what you should do with the papers, but basically this afternoon is a long alone time if you want. When you do the assignment that you have been tasked with and refresh your memory on the paper, so that later we can discuss about this in small groups in the small virtual room. For the paper, I tell you already right now, please pay attention to the question that they want to answer, the method that they have used, come up with at least an intuition if they could have used something different and do some different experiment. By now, you have all the means to actually come up with this type of things, and be critical. Is there things that really don't fit, don't make sense, or things that you would like to know further, curiosities, way in which you would address further questions that arise from the paper on? We will be calling you out and saying your opinion on this. So be ready to talk as well. And of course, at the end of this, one task that maybe was not in the assignment, write a short summary of the paper that you put in your lab book. Okay? Good. So any further